So now we're going to start working on the interior of the cell, which is much less complex than the exterior with all the different appendages. So the inside of a bacterial cell is filled with an aqueous water-based solution known as the cytoplasm. It is predominantly water, but it is mixed with things like sugars and amino acids that can be used as building blocks for things like proteins. But collectively, it is just an aqueous water-based solution that fills the entire inside of a bacterial cell. It's very similar to the cytoplasm that we have in our own cells. So all bacterial cells have to have genetic information in the form of packaged DNA that we know as a chromosome. So all bacterial cells will have what we call a bacterial chromosome. That is mandatory for all bacterial cells. It contains all the essential genetic information that is necessary in order for survival by means of things like growing and dividing and replicating. So all bacterial cells will have their bacterial chromosome. It's different than what we would expect for eukaryotic cells like human cells, whereas we have numerous chromosomes and they're all housed inside of that membrane-bound nucleus, and they're typically in linear formation. Bacterial cells often only have one chromosome. It is typically a circular chromosome. And it's hard to tell here, but if this were spread out, you could see that it was a circle. But they typically just have one circular chromosome. And it is important to note that it's not encased in a membrane-bound compartment, so it's not in a nucleus. It is simply just free-floating within the cytoplasm. Now, the region where the bacterial chromosome is within the cytoplasm is called the nucleoid, but it's not an encased membrane compartment as we would typically expect for a nucleus. So the bacterial chromosome present in all bacterial cells, it contains that kind of essential mandatory genetic information that all bacterial cells need in order to grow and divide. Some bacterial cells have an additional type of genetic information, and this would be housed on something called a plasmid. So a plasmid is optional. Not all bacterial cells have it, but plasmids contain non-essential pieces of DNA. So the plasmid would contain kind of the more advantageous type genetic information. The information that's not necessarily needed for survival, but can give bacteria some type of advantage in certain circumstances. So if a bacteria is resistant to an antibiotic, well, that information on how to become resistant to the antibiotic is likely found in the plasmid. Or if a bacteria knows how to produce a dangerous toxin, that information is likely held in the plasmid. That is not essential genetic information. It's not required for survival. However, it can certainly be advantageous and can give the bacterial cell some type of selective advantage. So, and typically when bacterial cells exchange genetic information through the pili that we were talking about earlier in the lesson, they're not going to exchange the bacterial chromosome because every bacterial cell has the chromosome. That's kind of the generic template. What they want to exchange is the plasmid. The plasmid contains the kind of really advantageous genetic information, like how to become resistant to an antibiotic. So what's typically sent to another bacterial cell in the pili is going to be the plasmid. So the next structure that we need to talk about is the ribosome. So bacteria have ribosomes just like we have, and the ribosomes still have the same overall function. They are involved in protein synthesis. So we can see a ribosome here reading this messenger RNA that it can eventually turn into a chain of amino acids or protein during translation, which we'll talk about in more detail later on in the semester. But overall, the ribosome is the site of protein synthesis. Ribosomes are composed of rRNA, which is ribosomal RNA, and then additional protein. They always consist of a large and small subunit. Typically, the large and small subunit are separate in the cytoplasm, but then they can come together to make a mature ribosome when they join up on the messenger RNA during translation. But there's always a large and small subunit. So one thing that you do need to be able to do is differentiate between the size of a prokaryotic ribosome and a eukaryotic ribosome. So prokaryotic ribosomes are typically smaller. The large subunit is actually going to be 50S in size, and then the small subunit is going to be 30S in size. And when you put the two together to make a mature ribosome, like what we're seeing here, it equals a total size of 70S. I know that doesn't seem like it makes sense because the two don't add together, but it's a measurement of density. So the prokaryotic ribosome is 70S in size. Eukaryotic ribosomes, like what we have, are much larger. Their large subunit is actually 60S, and the small subunit is 40S. And when you put the two together, it equals an 80S size ribosome. So they're much bigger than prokaryotic ribosomes. Which the size difference is important because a lot of antibiotics work by targeting bacterial ribosomes. But we also have ribosomes. So how is it that those antibiotics, when you take them, things like streptomycin, erythromycin, tetracycline, how are those antibiotics able to destroy bacterial ribosomes and destroy bacterial cells ultimately without targeting our ribosomes and affecting our healthy human cells? And the primary reason for that is the size difference. Those antibiotics are going to target the smaller ribosomes, the 70S size ribosomes. That will allow them to destroy bacterial cells without harming our eukaryotic human cells, which have the larger ribosomes. The next internal structure is also, it's an optional structure. Not all bacterial cells have this, and it is going to be the inclusion bodies. The inclusion bodies we would see right here, those kind of white structures that are found in the bacterial cells. Inclusion bodies are predominantly used to store excess nutrients or extra food. So if a bacterial cell ever finds itself in an environment where there's an abundance of nutrients, it is going to take in excess nutrients and store them in the inclusion bodies. That way, if it finds itself later on in an environment where there's not much food present, it can feed off of those excess nutrients in the storage for an extended period of time. So it's just a way to store extra nutrients. The bacteria can also pack gas into these inclusion bodies, which will give them buoyancy or the ability to float on a surface and form a film on water. So we often see that too, but the predominant function is food storage. And the last structure we've already talked about in our previous lesson was staining, but some bacteria have the ability to produce endospores. Not all bacterial cells can do this. It's typically just your gram-positive rod or bacillus-shaped bacteria that are able to do this. But bacteria that are able to produce endospores are able to survive in environmentally harsh conditions. And by that I mean they can survive in places where there may be waste buildup or toxic chemicals or maybe a nutrient depletion, or the temperature isn't a preferable temperature for bacteria. So they can survive in harsh environments. So basically how it works is if a bacterial cell is in a good environment, meaning it has access to moisture and nutrients and the environment's stable, it will be in its vegetative form. Again, I know typically vegetative we think is dormant, but we want to kind of reverse that thought process here. In this context, when we talk about vegetative, we want to think of the cells like vegging out, and they're eating and growing and dividing. So when they're in their vegetative state, they are metabolically active, which means they're growing and dividing. When they get put into a harsh environment, if they're able to, they will induce sporulation, which is just the process of forming an endospore. Basically, they make a copy of their genetic information, and then they surround it in a protein coat, and that structure becomes an endospore. and it will protect the genetic information for days, weeks, potentially even months, while the bacterium is in that harsh environment. During this time, the bacteria will be dormant. It will no longer be growing and dividing. It will just be kind of hibernating and protecting its genetic information until the environment stabilizes. Once the environment stabilizes and the nutrients come back and the moisture's back and the temperature's stable, the endospore will burst and the cell will be released in its vegetative form again, where it can continue to grow and divide until potentially the environment becomes harsh again, and then it will go back into its dormant form. And here we can see cells that have formed endospores. So these little green ovals inside of the cells are the actual endospores. And again, the older this culture gets, the rest of the cell will actually die off, and the endospore will be the only thing present. But that is going to protect the bacterial cell's genetic information for a period of time. And then if the endospore ever finds its way back into a good environment again, the cells will activate and become vegetative cells again. And this is a summary of this process and how it happens. I do not need you to memorize all the different steps. I just want you to have a general understanding that when cells are in a good environment, they will be in the vegetative form and they'll be growing and dividing. It's only when they get put into a harsh environment that they induce sporulation, which is the process of producing an endospore. I don't need you to know the specific steps, but basically they make a copy of their genetic material and they start to encapsulate it in various protein coats. Ultimately, that forms this structure that we know as an endospore, which just protects the genetic information for an extended period of time. So at this point, the cell is dormant and protected. Eventually, once the cell or the endospore gets back into a favorable environment, it will activate and go back into its vegetative form where it can continue to grow and divide. So it's very common for bacterial cells to kind of cycle between being in a vegetative state when they're in a good environment and an endospore state when they're in a hostile environment. They kind of go back and forth to survive for long periods of time. There are definitely very dangerous bacteria that produce endospores. Bacteria such as Bacillus anthracis, which causes anthrax, Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, and Clostridium difficile, which causes the disease that we commonly hear of as C. diff. All these bacteria can produce endospores. So that means they can survive in harsh environments like surfaces, which would lack nutrients, for an extended period of time. If any of these endospores got into your body, that is a good environment for them. They would activate, go back into their vegetative form, grow and divide, and obviously cause illness.